So welcome everybody to our first fully virtual afternoon in the science basement. Be kind to us. We've done some testing to see that the webinar will work fine, but we might still have some technical issues. So fingers crossed that everything will go well. Yes, so today our speaker is Outi Haapanen. Who's going to tell us about how to use supercomputers to simulate molecular dynamics. Before jumping into her very interesting talk, I would like to say a few words about the afternoon in the science basement for those of you that are new to this kind of event. So the Science Basement is an organization funded by PhD students, mainly from University of Helsinki, but also some from Aalto University. And we decided to create this association to offer a platform for young researchers to communicate their research to a general audience in English. And we offer different ways to interact with the general public. So afternoon is in the Science Basement is our series of public talks. They generally take place on Saturday afternoons, and they are live. So the audience comes to downtown Helsinki, and we have a speaker. There's like a public talk, and then lots of discussion that we hope to recreate here today. And then right now, our podcast team is launching the second edition of the Science Basement podcast. So stay tuned. I think next week will be the first episode. We also have different type of blogs. And we really encourage people that don't want to start their own science blog, but just try to post a little bit of content to contact us and maybe blog through our website. We also do some projects that to interact with schools and children. For instance, the Lego Lab is one of them. And we really enjoy mixing science and art. Some examples are the Night of the Arts that takes place in Helsinki. We joined it a few years ago, and we had a lot of poetry and music and science. And one of our members is also the person that was in charge of the climate wall in Helsinki. These very nice collaborations between climate scientists from the university and street artists from Helsinki. And here you can see different ways to contact us on the different social media platforms. And if you do want to be a speaker in one of the next afternoons in the Science Basement, please drop us an email. We are always happy to welcome different topics in science, not just we have a lot of biology and biomedicine and maybe more natural sciences, because most of our members are part of these fields. But we really hope to have more social sciences in the future and so on. So yes, looking forward to this. A couple of technical information. So if you have questions during the talk, since it's a little bit difficult to coordinate with the speaker, I would suggest that you post them in the Q&A, and then we will ask them at the end of the talk so that Outi can give her talk without having to monitor too many different windows. Now I would like to actually pass the stage to Outi, and while she sets up her slides, I think we have a short poll for you from the audience to know a little bit more. Where are you from, and how did you hear about us, so that we can improve the way that we reach you in the future. And yes, so I guess I'll stop sharing my screen somewhere. Yes, and now Outi, if you can share your slides and set everything up, and yeah, enjoy.
All right, so maybe Outi, just give a little moment for people to answer. Can you see the poll?
I think the poll is still there.
Yeah, so I'll let people answer maybe just for 2 minutes, if that's okay.
Okay.
Yeah, and if there's anything offensive in the poll, it's because I have a sense of humor that you don't like or something. So I, I apologize if I'm not funny, except to me. So it looks like we have 15, 20, almost 20 have already filled it out. So there's another, waiting on another 20.
And I guess we'll reopen the poll a little bit after the talk, if someone joins mid-talk and they still haven't filled it in.
I actually made a survey. So if you're answering these questions now, you can ignore later. When you close the Zoom webinar, you will get a prompt into your browser. So in case someone joins later and they were unable to participate in this poll. All right, I have almost 30. Can I get the last 11 people? You don't have to answer, but I would love it if you did. So it's been 2 minutes, maybe another 30 seconds, and then I will end the poll. So we have over 30 responses so far, and that's awesome. Right, no pressure, 10 seconds, a little bit of pressure, but don't feel bad. You can have a chance later. All right, 30 seconds are up. I'm gonna hit the end poll button now. Beep.
All right, then Outi, stage is yours.
Thank you. I want to make sure that you guys see my slides and you see my mouse pointer as well.
Yeah.
Great. Okay. So the title of my talk today is From Atoms to Supercomputers, What are Molecular Dynamic Simulations? My name is Outi Haapanen. I am a PhD student from University of Helsinki. And to start off my talk, I'm going to show the background image of the first slide. So as you are there listening to me and looking at the screen, this protein that you now see on the screen is actually functioning in your bodies. It is converting the energy that you eat in food into a form that is usable by your cells. And this protein is called the respiratory complex 1. We are going to return to this in a little bit more detail, and I'm going to show you an example of what molecular dynamic simulations have reached regarding respiratory complex 1. But before we get to more details, I'm going to start off by introducing the very basics of what are proteins. So respiratory complex 1 is a protein, and proteins are essential molecules in your body. As you know, everything in this world is made up of atoms, and so are proteins, so are you, so is the computer you're looking at. It's all atoms. So proteins are made up of atoms, which are grouped into smaller units called amino acids. And here on the left-hand side, you can see an amino acid. Let's look more detail to what is in it. So first, a pink box appeared, and this is what we call the backbone of an amino acid. What you see here is letters C and C alpha. These refer to carbon atoms. O refers to an oxygen atom, H to an hydrogen atom, and N to a nitrogen atom. And these atoms are bonded to each other to form the backbone of an amino acid. Every single one of the amino acids has this. However, as you may have heard, humans have 20 essential amino acids, and the difference between these 20 come from this guy, which is now in a blue circle, R. This is the side chain of the amino acid, and this is the one that changes when you look at different amino acids. The backbone is always the same, and the side chains vary. And then when you start forming your protein from these simple units, actually these side chains do not participate into forming the backbone of the protein. So the backbone of the amino acid, which is now in the pink, is the connecting item. You can see here now 2 units which are connected to each other. Here you can see the bond. And it is just the side chains. It's the main chains that connect. The side chains are merely on the side here. And the side chains, what are they formed of? Well, the same atoms as the backbone. It's just in a different order. So you still have the same types of atoms, carbons, nitrogens, oxygens, hydrogens, basically those that form life. And all proteins, the whole basics is about this. You have atoms which are connected to each other. And now you might wonder, like, this looks very different from what we saw before. Well, when we go back to looking at this, when we have a sequence of those amino acids that are together formed into a chain, they fold. They fold into a single unit or a collection of units, as in this case, which forms the whole protein. So you just have replicas of those amino acids in a certain formulations to make up whole protein. And here now, what you see is different ways to represent that. So you can visualize those atoms in different ways. You can see them as those letters. That is the very detailed view, seeing single atoms. But you can also view them as these white parts here, these curly ones. We call these curly ones helices. It is a name for a certain structure in a protein. It doesn't really matter what it looks like. It's just a way to represent it. But similarly, you see these blobs here in different colors of pink and purple and blue. They are actually the same thing. It's still the same atoms. Now I have just shown them so that they fill the space. And it looks like a blob instead of a curly helix like here. In reality, this guy here is the same structure as here. It is the same amino acids forming a chain and they have side chains. And this is what forms proteins. You're going to see several different representations of proteins during this talk, but it always, just remember, it's always based on the same idea. You have atoms connected to each other forming a big molecule. And that's just it. You can just show them differently to highlight different things. And one of the main things is, why is protein research so essential? Why do we need it? Well, the very simple answer is that nearly all the processes in the body involve proteins, and this includes transportation of smaller molecules, as I already mentioned, the conversion of energy in the food that you consume to a form that is usable by your cells as cell energy. Basically, everything that your body does most likely has some kind of a protein involved in the process. And as you can imagine, if something goes wrong, if you have in that amino acid chain, there's one wrong member. They are in the wrong order, for example. There's a mutation. This can cause several serious diseases. This includes different types of cancers, Alzheimer's disease, Parkinson's disease, etc. A big number of diseases. I just picked these ones because they are quite well known. And in case you don't understand how your protein works when it functions in the normal state, when everything is fine, it's going to be really difficult to find a medicine to fix that mutation. If the mutation has an effect on the function of the protein, if you don't know the original function, finding the medicine is much harder. So it is important to study proteins to possibly find cure for diseases that are lethal and serious. And this brings us to a very current topic of the coronavirus. Here on the right-hand side, you see a model of the coronavirus. You can see it is this blue blob. And what are these green things on the top? These are the famous spike proteins of the coronavirus. You may have read about them already in different media. You may have read about them in the news, because basically the vaccines are based on the spike proteins. That's how your body recognizes the virus. Again, it is an example of the essentiality of protein research. You need to have it. But now that you've seen a couple of examples of proteins, so you have seen the respiratory complex 1, which is related to the energy conversion, and you have seen the coronavirus spike protein, you might wonder, how did a protein from a human, from an animal, from a virus, how did it end up on the screen? How do you turn a protein into a computer model? Basically, I'm going to show you a very generalized way or the workflow of how a protein becomes a computer model. In reality, there are several different methods how it's done, but I'm not going to get into any detail. I'm just going to show a very generalized workflow for this. So of course, step number 1, you need to have a sample. Your sample can be from any organism you're interested in. It can be from a human, from a lamb, it can be from a cow, or even from a spinach. It can also be from different bacteria, from viruses, from yeasts, whatever tickles your fancy. And most importantly, you need to choose it from a tissue that actually contains the protein you want to study. So don't choose a tissue that doesn't contain it or contains a little bit only, because it makes life much, much, much harder. So when you have this sample of your protein, you need to prepare it. You need to remove all of the extra stuff that's not needed. You need to purify it, and you need to, depending on the method, you may need to be able to crystallize your protein, or you may need to plunge freeze it. And this is when it comes to about the methods. I'm not going to get into the details. You just need to prepare your sample to the point when you only have your protein and in the correct format. And this process is done in the wet lab and laboratory work. And sometimes this is the step that is going to keep you from finding out the structure of your protein, because actually you do not find a way to prepare your protein in the way that you could find out what is the structure. But let's assume that you did. Now you have a prepared sample. What you do next is you image capture it. Basically, you take your sample, you shoot it with rays. Depending on the method, it might be different kind of rays. And what you need to get is images of your protein from many, many angles, from so many angles. And by many images, I mean not just a few, not 2, not 3, I mean up to thousands of images. Because then, as you can see here, you have from different angles the same thing. It looks a little bit different. But when you have, say, thousands of these, and with computer algorithms, you combine those to form a 3-dimensional model called the density map. The map is now shown here in this blue mesh, as you can see. Here as well, you have a mesh, and you have a mesh here. That's what you get from the images. The next step is to fit a model in there. And remember when I talked about the amino acids, how they all have the same backbone, but different side chains. So you can easily find the protein backbones in your density map. And if you know, or when you know, in which order in your protein you have the amino acids, you need to start recognizing from these densities the different side chains, because they will look different there. And that's how you build a model of your protein. But what is the model actually? The final result is a PDB file of the protein. And now you remember, the protein, it consisted of atoms. And what is a PDB file? It's something super simple. It is a text file which lists every single atom of your protein. It gives it a number, as you can see here. It says atom, atom, atom. It gives your atom a number. You classify which element it is. So here we have, for example, a carbon, oxygen, oxygen, carbon, oxygen, nitrogen. It recognizes from which amino acid it is from, and it gives it a residue number. These don't really matter. Most importantly, you have atoms listed here, and for each one of these atoms, you have a coordinate: X coordinate, Y coordinate, and Z coordinate. So in 3D space, for each one of your atoms, you have a coordinate. You know where it's located in a three-dimensional space. And if you read this text file into a visualization program, it can draw you those atoms on the given coordinates. And then you can see your protein on the screen and you can rotate it around. And in fact, when I do molecular dynamic simulations, this is where my work begins. I take the PDB file of a protein. I look at it, I see where the atoms are, I see what the structure is like, and I start building a simulation setup. So into that PDB file, where I had the atoms of the protein listed, I will start adding stuff. And the reason is that proteins are usually not in a vacuum. So I will add, of course, first I will put the protein. I had the coordinates already for that. If it has some kind of small molecules related to it, I will add those. Again, just list of atoms with their coordinates. I will add a membrane, possibly. It depends. It might be that your protein is actually not in a membrane, but it's just floating around. And then I will add solvent, which is usually just water and sodium chloride. And I keep to the same format of file. I still have just a list of atoms with their coordinates. And if you think in a very simplistic way what a simulation box would look like, it is actually just imagine having a box where you drop tennis balls of different sizes. And some of these tennis balls are connected via sticks, and that represents the bonds. So you just have a box full of tennis balls connected via sticks. And in the actual format, this is just a file of atoms listed with their coordinates. And it is just that simple. You just took the protein from your sample and turned it into a listing of the atoms. And then I came into the picture and I put it into a box of water molecules. And then I want to start the simulation. And what is molecular dynamics, or MD? What is it based on? Again, quite simple. If you remember from high school, Newton's equation of motion states that force equals mass times acceleration. So when you have your coordinates and your interactions, so here you have now, here you have the list of atoms with their coordinates, and here you have how they interact with each other. We're going to calculate the force on each atom. So what is the force on that atom who it is interacting with? From those forces, we can, from this equation here, we can solve the acceleration because we know the mass, we know the force, we can calculate the acceleration. And when we know how that molecule, how that atom is accelerating, we can update the coordinates. And we have a new set, a list of the same atoms, but now the coordinates are just changed. And then we ask ourselves, did we simulate this for the desired number of steps or desired amount of time? If the answer is yes, we can view the evolution of the coordinates over time. However, quite often the answer will be no, we did not, because usually these simulation systems are run for millions to tens of millions to hundreds of million steps. So you just go through this loop over and over and over again. And because your system has hundreds of thousands to even over 1 million atoms, no laptop can do this calculation. And as a result, you use supercomputers. If I use my laptop, my run would never finish, basically. It's just, it would take too long. Even on the supercomputers, typically simulations take a few weeks to months until they're finished, because you're just going through this loop of calculating the forces and updating the coordinates and go again and again and again. So then why or why not should we use molecular dynamics? So the pro is that we can reach short timescales. So we can observe phenomena which takes place in nanoseconds or microseconds. However, on the other hand, we cannot study long timescale phenomena. So usually in experiments, the timescales that we're looking at are about a couple of seconds or up to say hours even. That's no way possible. In molecular dynamics, we're looking at the nanosecond to a microsecond scale. So this is parts of seconds, not even a full second. We can see the small, which means that we can look at atomistic details about interactions. If we have that mutation in the protein, we can see that how did it turn, what did it do, did it change its interaction partner, for example. We can see details like this. And these kind of details are sometimes very difficult to view otherwise. So we can model those interactions. However, if a reaction would contain bond breaking, which means that in your box of tennis balls, you would like to break one of those sticks connecting the tennis balls, yeah, that's not possible. With classical mechanics, you do not do that. Whatever bonds you have between your atoms, they stay for the rest of your simulation. You do not change them. It's not possible to do. And the definite pro of molecular dynamics with the short timescales and atomistic details is that we can reach phenomena and variables that are not available with experimental techniques. So we can look at things you cannot look in experiments, or you cannot look in the same way. We can also explain some phenomena which is seen in experiments, but they cannot see the details. Why did it happen? We can answer that kind of questions. However, the con is, as I already mentioned, we use supercomputers. So the calculations on the simulations, they are very computationally costly. They cost quite a lot of computing resources. Without supercomputers, it would be basically impossible to do the simulations. And very important thing to remember is that no model is good if it does not match the real world. The words by famous statistician George E.B. Box say, all models are wrong, but some are useful. And this also applies, I think this applies to all kinds of simulations and models. And this is why computational research is often, if not always, coupled with experimental research. It has to match the real world, otherwise it's useless. So now that we know the basics of what simulations are and what proteins are, let's look at the example of the respiratory complex 1. So we are currently looking at mitochondria. Again, if you remember from school, I think this has also become a meme online. Mitochondria are the powerhouses of the cell. And you can see in the schematic here that if we take, for example, a cow and we zoom in enough, we can see a cell. A cell is shown here. And then if you zoom into the cell, you can find these pink things here. You can see there's one, and there's another one. And these are called the mitochondria. Here I have a bigger image of that. And when we now go in and zoom to this wobbly part, to this wobbly part of the mitochondria, we can find respiratory complex 1. Does it look familiar? Yes, it's the same protein that you saw already before on the pink background. Now it's just fully shown as this space-filling model. So everything looks these kind of blobs on the screen. And there it is again, the same figure. So we're looking at the same protein we looked at in the beginning. But now let's look at a little bit more detail of its function. So let's change the view so that we actually see inside the protein. Now again, the same thing. You can see I changed the view a little bit. We still have the blobs and we still now have these curly bits here shown as they were before. But now I made it transparent so we see inside the protein. Let's see how it functions. Inside the cell environment, sorry, I clicked the wrong button. Inside the cell environment, we have these tiny molecules called NADH, which are just floating around. And NADH bumps into the protein and it gives it 2 electrons. These 2 electrons then move up to this yellow small molecule, which is shown here inside the protein. And this yellow small molecule inside the protein is called the quinone. And somehow when these 2 electrons land on the quinone, the protein is going to pump 4 protons across this part of the protein. And here is the funny thing. This reaction here, this electron transfer reaction, this happens much, much faster than this proton pumping. In addition, there is quite a bit of distance in between the quinone, which is here, to the furthest proton pumping site, which is all the way up to the other side of the protein. But yet somehow these two processes are coupled to each other. And the main question in respiratory complex 1 research is how this happens. How is that possible? So what I did, I simulated quinone starting from its original position here, as you see in yellow. I put it into a simulation and I decided to see what happens. So now we are looking at a close-up of that site. Here is the yellow molecule, as you can see. And this is now a simulation. Everything is wiggling around, everything is moving. The coordinates were updated over and over and over again. And what I saw that from its original position inside the protein, this small molecule, it actually moved through the protein to another site. So it moved from here to here. And as you may see now from this movement, it didn't go there smoothly. It wasn't just a whoop. Moreover, it took some steps. And when it took those steps, now you can see here the animation again. Look at it right here. When we now change the view so that I actually show this route as this beige kind of a dot, I saw that it made stops on the way. And the first two stops were actually already shown in structures. So my molecule started from here. It was a known site. It stopped the first time here, also a known site. But then it made 3 additional stops. Stops 1 and 2, these are known from structures before. 3, 4, and 5 were new. They were not known. When I presented my work in conferences, I got criticism and like very good comments and suggestions. And I got also questions like, why? Why would you have these 3 extra sites? But my simulations predicting these sites, it tickled in the minds of the structural biologists who started looking into their sample preparations. How could they change their sample preparation so that they might be able to either prove or disprove my theory of sites 3, 4, and 5? What ended up happening is that currently all of these 5 sites have structural support behind them. So there are respiratory complex 1 structures where quinone is observed at site 1, at site 2, 3, 4, 5. All of these are now found. So my simulations predicted that there is a possibility of having these sites 3, 4, and 5. And because I presented these results, because I showed them, it sped up the experimental research into finding ways to try to see the quinone at these sites. People would have not necessarily would have looked at them otherwise. So as a take-home message from my talk, I would like to say that molecular dynamic simulations are a strong predictive method, and they can help speed up experimental research on biological topics, as I showed in my example. If you have now questions and comments, it's time to start that. And I would like to thank all of my listeners.